Good morning, everyone. It's such a pleasure to greet all of you. I wish that we could actually meet. I could see your faces, but that's not to be. The pandemic still persists, and so we are being careful. Hopefully, next year it will be in person, or perhaps even the next mid-year meeting will be in person. I must thank those of you who have found the time to join this meeting. And I know that different sets of people will join at different points of time during the day and in the next 3 days. So I've titled my talk as Genes as a Guide to Human History and Culture. I'm primarily going to talk about human evolution, as you might imagine. And I'm primarily going to talk about reconstruction of human evolution using genes as a tool, as a guide. I'm also going to talk about one particular aspect of human culture that has been excavated using genetic data, or the spread of that culture has been identified using genetic data. So I'm going to also give you an example of that. So thank you very much for joining, and let me begin. No discourse on human evolution can be, or evolution for that matter, can begin without reference to Charles Darwin. So I'm going to first start with a slide or two of Charles Darwin. What did he have to say about human evolution? So in 1859, as all of us know, he wrote this magnum opus on the origin of species by means of natural selection, or the preservation of favored races in the struggle for life. In this particular book, he had almost nothing to say about human evolution. All that he said was a light will be thrown on the origin of man and his history. He, of course, went ahead and wrote another book in 1871, which he titled as The Descent of Man and Selection in Relation to Sex. And in this book, he made the point that man descended from the great apes. And he had a few points to say. He had a few important observations that made him conclude that man descended from the great apes. This is 1871. The kind of evidence that he provides might sound very funny or flimsy to us, but nevertheless, these were insightful observations at that time. And the arguments that he made is that there is a close correspondence in bodily structure between man and apes. The structure of the tissues and the composition of blood are similar. I don't think that he actually could have understood the composition of the blood at that time. He probably just looked at the color and maybe something else. I don't know that it coagulates and things like that. Men and apes have common parasites. This he certainly would have noticed. Process of reproduction is the same in all mammals. The embryo of man closely resembles the embryo of other mammals, particularly the apes, and it undergoes a corresponding order of development, and man possesses certain rudimentary organs, muscles, and other body parts. So essentially, these are observations on the phenotypic characteristics of man and apes that made him conclude that man descended from the great apes. He had no further arguments, and this is the set of arguments that he gave. And of course, today we know that whatever his arguments were, the final conclusion, final inference, has withstood the test of time. I'm going to tell you a story, essentially because the audience is kind of diverse from various disciplines of science. I'm not actually going to dwell on too much on the methodology. Essentially going to tell you the inferences that we have made using genetic data and use of mostly statistical methods in inferring from the genetic data. But I will give you just a little bit of a flavor of the methods, but won't talk very much about the method, and it will be more like in the form of a storytelling. So my story comes in, it's a two-part story. Part one begins 5 million years ago and ends about 150,000 years ago. And part two will begin around that time when part one ends, and it continues till the present day. The part one, which spans about 5 million years, I will complete in about 5 minutes. And the rest of the time, I will talk about part two, the last 130, 150,000 years or so. About 5 million years ago, a group of African apes split into two distinct species, one line that led to the humans, and of course, the species that the line that continued, the ape line continued, and that resulted in a formation of more species such as the bonobos. But the hominid line continues, and humans evolved in that particular line. We had a common ancestor, the great apes, the gorillas, chimpanzees, bonobos, etc. We had a common ancestor about 5 billion years ago. At this time, I will also emphasize that when I talk about these various dates, times, 5 million years, etc., you have to realize that these dates are not sacrosanct in the sense that there is a large standard deviation associated with these dates. And so when I say 5 million years, it could be 4, it could be 6, etc. So there is a large standard deviation associated with these date estimates. About 4 million years ago, a species that was on its evolutionary path to humans began spending most of its time on 2 feet, and that was the genus Australopithecus. A very famous member of the Australopithecus family is Australopithecus differences. This was, of course, excavated by Johansen in Africa, in Ethiopia, and this has been dated to 3.18 million years. So about 4 million years is when this particular species, Australopithecus, arose. Australopithecus is important because Australopithecus had a bipedal gait like we do. And the bipedal, how do we know that the Australopithecus had a bipedal gait? Again, all of these come from indirect evidence. So this is an animal that walked on a volcanic ash bed when it was still hot, and then it solidified and it's now a rock. And you can see the footprints on the rock. I can't, of course, read the footprints, but those who can vouch that whoever was the animal that walked on this rock bed had a bipedal gait. We can almost feel that these footprints could be our own footprints, bipedal footprints. But there are people who specialize, and there are people who can figure out that this animal had a bipedal gait. This rock has been dated to 3.7 million years ago, again around the same time when Australopithecines were ruling Earth. This was discovered not so long ago in 1975 by a family that has spent generations in Africa fossil hunting and reconstructing human evolution. This particular rock was identified, and the footprints were identified by Mary Leakey. The Leakey family has actually spent so many generations in Africa. So this is testimony to an upright gait. So around 4 million years ago, the species that eventually led to the line that eventually led to the humans had attained bipedality. Bipedality was very important in terms of human evolution because we had to survive. And the bipedality gave us our chance of survival to become much higher. And the reason being that, first of all, hands became free to use for manipulation. And one of the major manipulating things that we did was to throw stones at predators. And so we could survive longer. And these predators, then because of our ability to use throw stones, they went away without actually eating us. We also had a greater visibility over higher underbrush, which meant that we could spot predators from a larger distance, and then our ability to manipulate throw stones enabled us to ward them off from a larger distance. The third, and we wouldn't know whether this was a consequence or just a correlation or an association, around the same time, we also got dramatically larger brains. Whether it's due to bipedality, we don't know. And this dramatically larger brains meant that we could carry out more complex tasks. The genus Homo arose. We were talking about Australopithecines, Australopithecus as a genus. The genus Homo arose about 2 million years ago. Again, in Africa, Homo erectus, Homo ergaster, these are the kinds of species that were that existed around that time, about 2 million years ago. The hallmark was that we could use stone tools when we evolved. The Homo as a genus evolved. and Homo as a genus, members of the genus Homo, travel quite widely because we know that they were in Africa, and Homo erectus fossil has also been identified or found in Java about 1.8 million years ago. Once once the genus Homo appeared, it began to spin off new varieties of Homo. And these varieties have variously been called as robustus, habilis, ergaster, erectus, and so on and so forth. Eventually leading to the Heidelberg man, the Neanderthal man, and ultimately the Homo sapiens. We belong to a subspecies of Homo sapiens called sapiens, so we call ourselves Homo sapiens sapiens. So that's what the human evolution was in the last or evolution in the last 5 million years was that led to this particular line of descent, eventually leading to us, Homo sapiens sapiens. Incidentally, the gorilla and the chimpanzee and the great ape line continues because we know that they continue to evolve and they continue to exist. After we evolved, and this is really the branching of the various kinds of species that arose from the genus Homo. We were talking about Homo erectus, and then there were other branches that came out of that particular basal trunk, and these were antecessor, erectus, and so on and so forth. As you will notice, that many of these branches actually died out. Many of these branches are dead branches, and they did not evolve any further. There are various reasons why branches in evolutionary trees die. We won't get into that. What I do want to point out is that from that basal trunk arose another particular species that we called as Heidelbergensis. This is the Heidelberg man. And the Heidelberg man gave rise to 3 different branches, one that led to anatomically modern humans, the Neanderthal man, and the Denisovans. As you can see from the branches, the Neanderthal branch and the Denisovan branch, those 2 branches fuse with the human branch, and we will have a little bit more to talk about this. In terms of dates, so that's the Heidelberg man, and looks like one of us almost. The split from this line, from the Heidelberg man line, was about 2 that led to the Neanderthal man and the Denisovans was about between 300 and 400,000 years ago. And after a bottleneck, the one that led to the humans was about 150,000 years ago. So anatomically modern humans arose about 150, 130,000 years ago. So that's the 5 million years that I was going to talk about, and that was the part 1 of our story. Part 2 of our story begins now, and part 2 of our story begins with the modern humans. We are less heavily built than the great apes, than our ancestors. We are more mobile. We have a higher cognitive flexibility. We arose in Africa about 130,000 years ago, and we will have a little bit more evidence to show that we arose in Africa. Now there are multiple lines of evidence that actually point to humans arising in Africa, but I will provide some genetic evidence. We evolved somewhere in Africa, possibly in the Ethiopia-Kenya region. We were a small group of people. We were hunter-gatherers, and they look like us. Our ancestors, immediate ancestors, really look like us, with a high forehead, sharp chin, light, graceful bodies, and this is one of our great ancestors. It's of course a reconstruction, but based on a lot of fossil evidence. And around that time, many other human-likes, what are called archaic humans, lived elsewhere in Africa. When we actually arose, when modern humans arose in Africa about 130,000 years ago, there were many archaic humans that lived elsewhere in Africa and also in other places such as Asia, Europe at that time. They looked noticeably different from us. They had a low forehead, heavy ridges around the eyes, etc. And there are reconstructions of these skulls and bodies. And there were about a million of these archaic humans when we evolved. And this is, of course, these are all estimates. But anyway, the last of them, or after about 30,000 years, there are no fossil remains of any of these archaic humans, and it's estimated that there were about 1 million of them when we evolved in Africa. We will never know how they vanished. Perhaps we over-reproduced, outnumbered, and killed them. But this is a perhaps. Will never know. We likely that was the most likely scenario, that we over-reproduced, outnumbered, and killed them. But today, these belief from various kinds of evidence is quite different, and I will present to you that the evidence. Today, modern humans, anatomically modern humans, there are about 6 billion of us, and each one of us descended from a very small group of anatomically modern humans. Millions have lived and died, but fossil remains don't exist. Only a few fossilized remains exist. So to be able to construct, our footprints, our evolution, our migration trails, etc., imposes a very high burden of evolution, primarily because scanty fossilized evidence remains. And it's hardly for modern humans. We usually have very fragmentary evidence, one broken bone, a broken skull, and so on and so forth. So with this kind of fragmentary evidence, it imposes a heavy burden of speculation. Is there any other evidence that we can, or any other body of knowledge that we can fall back on? Well, another source of evidence is DNA. Up until recently, it was not easy to identify or to isolate DNA from fossil remains or from individuals who died many years ago. But even from contemporary contemporary DNA evidence, DNA sampled from contemporary humans living in different parts of the world, we are able to reconstruct human evolution using various kinds of statistical knowledge primarily. So let me just describe to you our bread and butter in terms of reconstructing human evolution using genetic data. So what we do is we usually a blood sample is taken, and from the blood sample we isolate DNA, and then we sequence DNA. Earlier, it used to be in bits and pieces. We would see, because of the costs involved, costs and limitations of the technology, we would sequence only portions of the DNA, small portions of the DNA. But today we are able to sequence the whole genome, which is about whole human genome, which is about 3 billion nucleotides, and we can do this at a reasonable cost. And therefore, because costs have fallen, we can actually sequence multiple individuals. But why do we have to sequence multiple individuals? The reason being that not all of these sequences will look the same. There are particular positions where not all of us will look exactly. Not all of us will have exactly the same sequence. So each row here represents an individual, and as you can see from that arrow, if you go along the column, there are changes. It's predominantly G, but there are also changes like another alphabet T, and these are called mutations. These mutations arise, and they get distributed over into different individuals. So those are positions which are variable. These positions are called polymorphic positions. The positions that are not variable are called monomorphic positions. Polymorphic positions have a lot of information about drawing inferences on human evolution or evolution of any species for that matter. The monomorphic positions don't contain as much information, but together with the polymorphic, these are called polymorphic loci or positions. Together with them, it also can provide some bit of information. Won't get into the details of it. So both polymorphic and monomorphic positions matter. Monomorphic positions are much less informative than polymorphic positions. So we are looking at these DNA sequences, and we want to reconstruct the history of DNA sequences. So essentially what we do it in two steps. We are reconstructing the evolutionary history of a genus or of a species, and we have sampled a whole set of DNA sequences from contemporary living individuals of that particular species. And now the first step is to be able to identify is to be able to reconstruct the history of those DNA sequences. Once we reconstruct the history of those DNA sequences, we partition or we group these DNA sequences into populations and see what kind of relationships are there between populations. So conceptually, it's really a two-step process. So I will give you a flavor of how we reconstruct the history of DNA sequences. These DNA sequences have been drawn from individuals. So eventually, all of these DNA sequences will root back, will go back to one single DNA sequence from where all of these DNA sequences have arisen. And that's called that DNA sequence belongs to the most recent common ancestor of all of the DNA sequences that we are looking at. And we can estimate times, etc. So here is an example of 22 individuals. So 22, let's say haploid. We know that our chromosomes are paired, etc. But allow me for a second, or for a few minutes, to imagine that we are not paired chromosomes, but unpaired chromosomes, so-called haploid individuals, such that there is only one DNA that represents an individual. So these are 22 DNA sequences drawn from 22 haploid individuals, and we want to reconstruct their evolutionary history. Of course, we realize that this is in contemporary times, and we are actually looking back in time. However, evolution is a forward-looking process. So in some ways, we are tracing back time, but eventually we will have to look forward in time in order to understand how this evolution has taken place. So anyway, we look backward in time, and this is what we do. We look backward in time, and these 22 DNA sequences have arisen from 18 DNA sequences. And why is that? Because some individuals would not have procreated, would not have reproduced, they have no children for the next generation. Some individuals would have produced more than one child. So that's how you see that there are these green ones who do not have descendants in the next generation, and there are some where there are two arrows, meaning that there are two descendant DNA sequences from one DNA sequence. Oops. So these 18 ancestors would lead to 16 ancestors, and we can go on and on. And eventually, if we remove all of the dead branches of the tree, we will go back to one single common ancestor, and that's called the most recent common ancestor. This DNA sequence of the most recent common ancestor led to these 22 DNA sequences that are present in these 22 individuals, and these 22 individuals don't look all alike. This whole thing was actually inspired. The methodology was provided by J.F.C. Kingman, and this is called a coalescent process, because we are looking at contemporary DNA sequences, and contemporary DNA sequences are coalescing back to one DNA sequence, joining back to one DNA sequence, and that process is called a coalescent process. The process that underlies reconstruction of these DNA sequences is popularly known as the Kingman coalescent. I don't have the opportunity to provide you with details of the Kingman coalescent, but I want to point out two things. The Kingman coalescent rests on a Markov chain, and again, I'm not trying to reach out to every one of you, but I do want to point out that it is the biology is legitimately a Markov chain, for the simple reason that if I consider my own DNA is completely determined by what my parents had, and my grandparents don't matter. It's only my parents. So it's only one-step transition that matters, and that's what, given this one-step transition, the previous generations don't matter at all, and that's the reason why it's a Markov chain. Usually, time is measured in discrete generation, as discrete random variable. But for the Kingman coalescent, because these generations oftentimes don't synchronize, it's a continuous time Markov chain is used. Again, as I said, that I'm not going to provide you with even the equations of this stochastic process, but it's essentially a Markov chain that leads to this particular reconstruction of the histories of these DNA sequences. It's quite interesting that the way that Kingman formulated the coalescent process, it actually factorizes into two components. one of which is the pure birth process, and the other is a discrete time jump chain. Again, these are technical, probabilistic, or stochastic process phenomena. I'm not going to get into the details, but all that I want to tell you is that this happened not so long ago. Kingman coefficient was formulated and published in 1982. So all of these kinds of things that we are able to do now, which is completely dependent on the Kingman coefficient, happened only between from about the 1980s. So we are a young breed of people who are using these kinds of methods in order to reconstruct human evolution. Like I said, that these 22 DNA sequences don't look all the same because mutations have arisen. And so what one needs to do is to sprinkle mutations as they had arisen on this phylogenetic tree, on this evolutionary tree. So so let's say that that's a mutation that had arisen long ago, and all of the descendant DNA sequences therefore will carry this mutation. On this, you could have other mutations that arise. So these are different kinds of mutations that are arising. And on the background of one mutation, you could have another mutation that arises, and so on and so forth. So as time evolves, these DNA sequences become more and more distinguishable, distinct from each other. And these, as humans have evolved, or as any species has evolved, they usually group themselves into smaller number of individuals that are called populations or subpopulations, and these DNA sequences are not uniformly spread across the subpopulations. So if we essentially, if we group these DNA sequences into subpopulations, these 22 individuals belong to 3 different populations. And as you can see from the history of the DNA sequences, the DNA sequences are not uniformly spread across the 3 populations. Population 1 has more of those blue sequences, and population 3 has more of the red sequences, etc. So the characteristic features, genetic features of these DNA sequences that are embedded or encased in these populations is a result of human evolution. And by reconstructing the histories of these DNA sequences, we can actually infer about the relationships, evolutionary relationships among the populations. So this is the way that we just giving you a flavor of what we do using these DNA sequences. We try to understand relationships among populations by understanding relationships among the DNA sequences, and these DNA sequences being encased in certain populations, contemporary populations. We do much better. It's not just looking at pictures. We can actually estimate times. Again, estimation of time depends on certain kinds of models, and these models will usually have certain assumptions. Some of these assumptions are flexible, meaning they can be relaxed to identify what kind of impact they will have on the time estimates. So anyway, this, like I said, that two DNA sequences will join to one, and that's called the first coalescent event. So as we move from contemporary to looking at the past, there will be various kinds of coalescent events happening, and we can estimate the times of these coalescent events using the Kingman coalescent and derivatives of the Kingman coalescent. I'm not going to get into details of this. It will form an exponential distribution with certain means and standard deviation, and we can estimate the heights of the trees and therefore the times. Just to give you qualitatively what happens, we reconstruct using the standard procedures of phylogenetic analysis, reconstruct the relationships among these DNA sequences, and ultimately look at these trees and find out and draw certain inferences about what may have happened. So I'll just give you these trees depend on constancy of population size. These in the total size of the population obviously changes, but if it is constant, what happens is that most of the coalescent events will occur near the base of the tree, meaning that most of these coalescent events will occur in the recent past. If we contrast this with, let's say, an exponentially growing population, which is for humans, it's not completely exponential, but that's a fairly good approximation to how we have grown. Most coalescent events will occur in the distant past than near the base of the tree. I'm giving you these in some qualitative sense, and as you can see, from the trees themselves that the two sets of trees that I just showed you, they are qualitatively different. But again, you can estimate times, you can estimate the parameters of the exponential distribution using this. Those are technical details, and we use all of that in order to be able to in order to draw inferences about how about our evolutionary history. So I'll stop here with respect to paradigms and methods, and I really want to go back to my story, which is to trace back human evolution using genes and DNA. So we came out of Africa, and again, I will provide a little bit more evidence as that it is consistent with our evolution in Africa, genetic evidence consistent with our evolution in Africa. So we came out of Africa and we saw that we were not alone, and I've already alluded to the fact that there were human lights that were traveling or roaming around the streets of Asia and Europe at that time. So we came out and saw that we were not alone. We met at least one other or two other hominid species who are our cousins on the European landmass. One of them is the Neanderthals, and there are multiple fossils of Neanderthals that have been identified, and they're quite well preserved. And so there were bits of muscle that could be extracted from those Neanderthal fossils, from which DNA could be isolated and DNA could be sequenced. So we have multiple Neanderthal DNA sequences now. We also know that we came out and met another cousin of ours, which was found in a who was found in a cave in Siberia, and a small finger bone was found, from which DNA was isolated and compared first with the Neanderthal DNA, because the proximal conclusion was that whoever lived in this cave, whoever had this finger bone was a Neanderthal. But that was very quickly disproven because of the extent of differences in DNA between the Denisovan, the DNA sequence found in the Denisovan cave and the Neanderthal. So this cave is called Denisova, and that's why this individual has been called Denisovan, and Neanderthals below. So Neanderthals and Denisovans are not the same. The individual who lived in this cave, in Denisova cave in Siberia, was completely different from the Neanderthal. So there must have been another cousin that we were dealing with. So these sequences were compared, and we can identify what proportion of the DNA of a contemporary DNA has come from other putative DNA, ancestral DNA molecules. So since the Denisovan DNA, the Neanderthal DNA, and multiple human DNAs were already sequenced, we could estimate the proportion of admixture, and what was quite evident is that the Neanderthals admixed with the Denisovans and provided their bits and pieces of their DNA to the Denisovans, and the Denisovans and Neanderthals both contributed to the human DNA. When we looked at modern human DNA, there was also some bits and pieces that could not be traced back to the Denisovan or Neanderthals, and so there was this possibility of a potential unknown hominin. So there probably are more Homo species, human-like species, that are to be discovered and have not yet been discovered because their fossils may not have been preserved very widely. So I have a caption that says they made love, not war. And the reason why I say this is that please remember that earlier I mentioned that the Denisovans and the Neanderthals, their branches fused with the human branch, unlike other branches of this human evolution tree, which became dead branches. At that time, before all of this evidence came along, the favored hypothesis was that we outnumbered, humans outnumbered, we over-reproduced, outnumbered, and we killed the Denisovans and the Neanderthals. But what we now see is that's not what we did. If we did do that, then their DNA could not have contributed as much to our DNA, to modern, anatomically modern humans' DNA, and therefore we bred with them, we mated with them, we reproduced with them, and that's how we know that we made love and not war. There are, like I said, there are other potential unknown hominins as well. Homo sapiens is, of course, reproductively more successful than both the Neanderthals and the Denisovans. And one reason why this may have happened is our ability to use tools much better than the Neanderthals and the Denisovans. In the larger context, we outnumbered them for sure, but not by killing them, by absorbing them into our own species. Like I said, that if you look at the human DNA, you will find Denisovan and Neanderthal DNA in our genomes. Similarly, Neanderthal DNA contains Denisovan DNA and also bits and pieces of human DNA, and so does the Denisovan DNA. Also, when we were studying the Andamanis or Jarwas and Onges, tribal populations of Andamans, and we sequenced them, we found that there were certain small sequences in the DNA, small pieces in the DNA sequence, which did not trace back to the Neanderthals or the Denisovans. And so what we tried to do is to try and date these, and those are the yellow pieces that have appeared. We asked ourselves, where did these yellow pieces come from if they didn't come from if they cannot be traced back to the Neanderthals or the Denisovans. So in our quest to understand that, or to answer that question, we looked at multiple populations of mainland India, and we did not find these yellow pieces anywhere in any of the humans who live on mainland India right now. We then started to collaborate with some people in Southeast Asia, and there are many tribal populations in Southeast Asia, in Vietnam, in Thailand, and so on and so forth. And what we found was that, okay, so before I tell you what we found in Southeast Asia, let me also tell you that since we did not find these in mainland India, we thought that it would be interesting to find out where the time to which these yellow pieces traced back. And indeed, what we found was that these yellow pieces, if we looked at the mutational spectra and when they had arisen, it was around the same time that the Neanderthals and Denisovans lived. So these are ancient pieces of DNA and didn't arise in recent times. When we looked at South Asian tribal populations, and indeed what we found was quite interesting, that many of the Southeast Asian tribal populations did have this yellow piece, which essentially meant that there was another potentially unknown hominin who walked around the streets of Southeast Asia, forests of Southeast Asia, and mated with humans around that time and contributed the DNA. No fossil evidence of that particular unknown hominin has been found. Maybe it's not been preserved well, but the DNA evidence points to this unknown ancestor being present and having contributed bits and pieces of their genomes into the human DNA. When we completed this work, we published this work some 5 years ago in Nature Genetics. Won't explain this picture at all, but what I want to tell you that we published in 2016, and in the same year, during the American Society of Human Genetics meeting, there was a group from Melanesia who actually studied tribal populations of Melanesia. And again, I'm not presenting their data, but their conclusion was that they did a genetic study of these ancient Melanesians, and they also identified that these ancient Melanesians interbred with the mysterious hominid, and the way that they had drawn this inference was very similar to the way that we had drawn this inference using genetic data or DNA evidence. We are unusual animals. We have a wide geographical distribution, and we have adapted ourselves to a wide range of environmental conditions. We are peculiar in that we choose mates from within our own group, and these groups are usually defined by certain kinds of cultural context, cultural traits, and that's how we form our groups. And these cultural traits, the most dominant cultural trait is language, which has also evolved, but as a result of the fact that we mate within our own group, genetic variations that arise within the group tend to remain localized because we are not freely intermating. So the genetic variations that arise within the group tend to remain localized within that group and not move on to another group through mating. And therefore, because of this phenomenon, various population groups tend to retain some measure of genetic distinctiveness. And our entire bread and butter actually exploits this fact that the groups, the genetic variations that arise in the group remain within the group, and we retain some degree of genetic distinctiveness. There are, of course, we are not freely intermarrying primarily because of cultural differences, but also physical differences. There are geographical barriers that are a major barrier to admixture. Imagine, if you will, that there are two population groups or two subgroups of a species that are living on two sides of the Himalayan mountain chain. There is hardly any possibility of their mating, interbreeding. I talked about cultural differences being a barrier to admixture, and similarly language differences are also a barrier. So there are multiple barriers to admixture, and therefore we group ourselves in this way. When we look at these genetic data, again, because of this phenomenon that anything that arises in a group tends to remain localized, if you have a population group that arose a long time ago, that particular population group would have accumulated a large number of genetic mutations that would not have been passed on to other more recent population groups. And therefore, if we look at the other side of the coin, where we estimate the genetic diversity of populations, we can conclude as a result of what I just mentioned, that those populations that have a higher level of genetic diversity are older populations. So people had early on, researchers had estimated using various kinds of genetic information, what kind of genetic diversity is there in populations of Africa, Asia, Europe, etc. And what we find from this graph, the x-axis is essentially a measure of genetic diversity. What we find is that the genetic diversity in Africa is very high, and followed by Asia. So we have, the African populations are more diverse and therefore older. And so this is consistent with the view that humankind arose in Africa and populated other parts of the world later. Give you a more direct evidence, at least the pattern. These are signatures. I'll just call them as signatures. Technically, they're called haplogroups, but I'll, for the sake of this talk, I'll just call them as signatures. These signatures, A, B, C, D, E, etc., are all genetic signatures that are present on the Y chromosome. Y chromosome in humans are, as you know, are only present in the males. So as we look at the distribution of these Y chromosome signatures, we are actually looking at the distribution of or the migration or the movement of humans as they carry these signatures. The signatures have been also arranged in such a way that the older signatures are on your left, A, B, C, D, etc. are older signatures. The newer signatures are on your right. What we now did, a large number of us collaborated, identified these signatures in various contemporary population groups from across the world, and we mapped them using pie charts, the proportions of these various kinds of signatures in populations of various continents, and this is what we find. I'm not going to leave this on for a long period of time. I will summarize what you would see is what I've seen. Gazing at this for a long period of time, is that the oldest signatures are in Africa. The newest signatures are in the New World. Humans crossed the Bering Strait and populated the New World, the Americas, and the middle-aged signatures are all in Asia. So this is consistent with humans evolving in Africa, coming out of Africa to populate parts of Asia and Europe, and then moving on to populate the New World. This is this is a male population movement. Skip this slide. This is female population movement as enshrined in what's called the mitochondrial DNA. Again, it's exactly the same thing, except that the signatures are named in a funny way, but doesn't really matter. And what you see is exactly the same kind of conclusion that I've drawn for the male population movements. The oldest signatures, oldest mitochondrial DNA signatures are in Africa. The newest ones are in the New World, and the middle-aged ones are in Asia and Europe. Both male and female signatures bear testimony or are consistent with the hypothesis that humankind arose in Africa and moved out of Africa to populate other parts of the world, most recently the New World. We were interested in focusing or placing Indian population groups in the context of Asia. We knew that from previous studies, we knew that humankind came out of Africa. Some of the places that they occupied were in Asia. We were interested in figuring out, relative to the other Asian locations, when did humans get into India? And so we again collaborated with a large number of human geneticists and essentially came to this conclusion. The root of the tree is at the bottom, the left-hand side is at the bottom. And all of these are contemporary human populations sampled from various parts of Asia. And this is a reconstruction using DNA data of the various populations that reside in Asia. And what I'm showing you is the arrow is that there was an early divergence of individuals who belong to population groups of India, which essentially means that India was occupied early on in the evolution of humans or in the migration of humans after humankind came out of Africa. And what we also see is that we can estimate looking at the nature of the DNA sequences, we can ask the question how many ancestral components are there or are there in the contemporary populations of India, and we find this is color coded. We find that there are 2 ancestral components. If you look at the whole set of Asian populations, our estimate is that there are 11 ancestral components in Asia, 2 of which are in India, and these 2 ancestral components arose, diverged soon after humans came out of Africa, and the first place that they occupied was India. If you look at ethnic composition of India, we have a large number of tribal populations, caste populations, religious and migrant groups, and they are culturally divided by the complexity of their social organization, by the nature of the language that they speak. Most people of India speak 4 different kinds of languages: Austroasiatic, Dravidian, Tibeto-Burman, and Indo-European. Indo-European is spoken in the north, Dravidian is spoken in the south, Tibeto-Burman languages are spoken in the northeast, and Austroasiatic languages are spoken exclusively by the tribals, and these are tribals of central India primarily. And these numbers of tribal groups like 450, etc., caste and subcaste groups, religious and migrant groups, these have been estimated by the Anthropological Survey of India using a nationwide survey of their culture, of their physical dimensions, and so on. We again, in our quest to reconstruct populations or understand the relationships among population groups of India, we sampled a large diversity of individuals from various populations of India, and those are the places from where we sampled them. And these populations are both tribal populations and caste populations at different hierarchies. We sampled about 600 of them. Of course, that's not even representative of all of India, but anyway, the costs are, even though the costs have come down, the cost of whole genome sequencing is still quite high, and we were able to sequence 600 DNA sequences, and we have reconstructed relationships among Indian populations using these 600 DNA sequences. One of the first things that we asked is there Neanderthal admixture in Indian population groups? And the answer is yes. It varies between 2 to 2.5%. So that's the extent of Neanderthal admixture in Indian. We then ask the same question: do we find Denisovan admixture? And again, we do find Denisovan admixture, and that's about 1.1 to 0.4%. So the extent of Neanderthal admixture is orders of magnitude greater than the extent of Denisovan admixture among Indian populations. Interestingly, by the way, there was no geographical patterning of admixture either for the Neanderthals or for the Denisovans. So it's not like South Indian populations had a greater level of Neanderthal admixture than North Indian populations. We couldn't find such kind of geographical patterning. However, with respect to the percent of Denisovan admixture, even though the percent of Denisovan admixture in the genome is quite small, but there was an interesting patterning, as you can see, that as we move from the tribal populations, which have the highest amount of proportion of Denisovan ancestry, to the more recent populations, the upper caste populations, there is a gradation, and these differences, some of these differences are statistically significant. We published this a couple of years ago in Nature. So the last question that we asked is how many footfalls, how many distinct ancestral types are there in India? We addressed this in our paper in Science that where we collaborated with a large number of researchers from various parts of Asia. There, the number of Indian populations that we were able to sample was very small, and we estimated that there were two ancestral types. I'm going to skip this, and I'm also going to skip all of this and go over to this slide, which essentially shows that there are 4 different colors that you see, and these different colors represent 4 ancestral populations. So let me explain this graph to you for a minute. On the x-axis, there are dots, each dot representing an individual. There are so many of them that you don't even see those dots. On the y-axis is the proportion of different ancestries, and we estimate that there are 4 ancestries. These are 4 different ancestries. So there are some populations, some individuals who are completely pure, meaning that they have their genomes comprise only one ancestry, and there are individuals sort of in the middle of the picture whose genomes are checkered with 4 different ancestries. Can we identify these ancestries with contemporary contemporary traits of human populations? Yes. The green ancestry primarily is an ancestral population to the Indo-European speaking populations of India. The red one is an ancestral component of the Dravidian populations, etc. So essentially, we've identified 4 ancestral components in the in populations of mainland India, and these can be identified with the various languages that these people speak. We published this again 2 years ago in PNAS. I mentioned that I'm also going to provide you an evidence of a cultural trait being inferred, movement of a cultural trait being inferred from genes. So a cultural trait, which is, let's say, a technology which evolves as in a specific culture, human cultural context, moves from one place to another place, and that movement can happen in one of 2 ways. It can happen through diffusion of ideas, idea of the technology and how you use that technology. That can happen by word of mouth, or there may be people who actually take the technology to another place and teach the local people there how to use that technology to their benefit. So there are 2 ways in which an idea can move, or a cultural trait can move, either through cultural diffusion, no movement of people involved, or demic diffusion, where there is movement of people involved. One of the major cultural events that have taken place in human evolution is farming. Farming as a technology is a cultural trait that has moved from what's called the Fertile Crescent region, which is Syria, Lebanon, Turkey. That's where farming arose in its present form, in its modern form, and has spread throughout the world today. Was it through movement of people, or was it through movement of ideas? We concluded that it is through movement of people, because if you take one particular signature that's in very high frequency in the Fertile Crescent region, you find, this is a heat map, you find that from that it gets diluted. Now, this is, and it gets uniformly diluted as you move from the Fertile Crescent region away from the Fertile Crescent region. If it was through word of mouth, this particular gradation would have been very patchy. and probably there would not be any such interesting clinal pattern to this distribution of this particular signature. We extended this to India. Skip this. So this is essentially using Y chromosomes. Farming was probably male mediated, and so this is again a relationship of Y chromosomes that we identified from various population groups of India. And if you look at the distribution of this particular signature, haplogroup J signature, what you find is that it moves from the fertile crescent region into India, and there is a very nice gradation, as you can see from this heat map. There is also another cline that you can see that's sort of towards the northeast of India, and most probably that's rice that came in from China into India. So we are talking about cereals, wheat, etc., from Middle East, from the Fertile Crescent region, and rice from China that came into India. I don't have the time to show you now. There were rudimentary forms of agriculture also in India, but those rudimentary forms of agriculture traveled within India but did not travel outside of India. And those rudimentary forms of agriculture actually preceded the modern farming. So modern farming in its present form, so genes can also provide guidance to the migration of cultural ideas and cultural traits across across geographical space, across various population groups. So I will stop here. I have reconstructed human evolution to the best of my abilities using genetic knowledge or genetic data, and also relationships among Indian population groups in relation to the ancestral Denisovan, Neanderthal, etc., as also the contemporary relationships among contemporary population groups, identified how many ancestral types may there have been who contributed their genomes to the contemporary populations of India, and have also been able to show you that if we consider farming as a cultural trait, then genes can also provide guidance to the dispersal of a cultural trait. As we deal with these genetic data, human history shows that genetic data have been very widely misused for purposes that they were not intended to. Collection of which was not intended to use the data in such forms of racial discrimination, etc. So I'll come to my last slide, which I think is my most important slide, which is a passage which I'll read out to you, which is a passage from the African American poet Maya Angelou, and she wrote this little book called book of essays called "Wouldn't Take Nothing for My Journey Now," and I have to read this out to you because that gives me a great satisfaction. It's time for the preachers, the rabbis, the priests, and the pundits, and the professors to believe in the awesome wonder of diversity so that they can teach those who follow them. It is time for parents to teach young people early on that in diversity there is beauty and there is strength. We all should know that diversity makes for a rich tapestry, and we must understand that all the threads of the tapestry are equal in value, no matter their color, equal in importance, no matter their texture. Thank you very much for your attention.
Thank you, Partha, for that very enjoyable address. A couple of questions. I'll read them out. One is from Deepak Pental, who talks about absorption of Denisovan and Neanderthals is a romantic idea. He says, in reality, a tribal mentality exists in Homo species. The situation of one tribe stealing the women of another tribe has been common in human tribes, including modern tribes.
Deepak, the major problem is that these are being identified from not so well preserved specimens, and it's very difficult to, even though there are thousands of mitochondria, the mitochondrial genome is very small. And so it's not been possible to isolate mitochondrial DNA. Only nuclear DNA is what is possible. So I don't know. I wish that mitochondrial DNA and Y chromosomal DNA could be simultaneously studied in the Neanderthals and Denisovans, but that has actually not been possible.
Right. So then you have a question from Satyamurthy who says, how come the Americas are missing in the picture of old times? And that Lalji suggested that the Jaroas come from Africa.
Okay. So three things. One is that how come the Americas are missing from the picture of old times? The Americas have been populated max 20,000 years ago. And humankind came out of Africa, they walked and they crossed the Bering Strait and moved into the New World. So there was no ancient peopling of the Americas. It was only about 25,000 years ago at the max. The Jawas, again, have certain genetic characteristics that go back to Africa, but whether they came from Africa for sure is it's still is still debated. Their physical features, of course, very well correspond very well with the Africans, but in terms of rooting the Jawas and the Conges, their DNA sequences with Africans, it's cannot be done. There are certain traits in the DNA that root back to Africans, but when we do the whole genome sequencing, we don't find that corresponding to the such good correspondence to the African genomes.
Okay, so then you have the last question from Yogita Adlaka. And the question is asking, it is surprising why humans have arisen early in Africa and not in other parts of the world. Were conditions in Africa better than other parts for survival?
Well, we don't know. Again, $1 million questions. This often comes up. Why did humankind evolve in Africa? As a matter of fact, not just humankind, but also various other genera evolved in Africa. Why Africa is the cradle of so much of biological evolution, one never knows, but we can probably never answer those. The conditions, there were similar conditions, climatic conditions and physical conditions were similar in many other parts of the world, but why they arose in Africa, we never know. No answer to this question.
Okay. I just, you've got lots of congratulatory messages from Madhu Dikshit and N. Satyamurthy and Subir Majumder and R. Ramachandran and lots of other people are saying that your talk was superb, and I think we all agree with that. So thank you very much, Partha, for an excellent address.